Hello S3, today we are going to start the second subtopic of Unit 1, Transport across the cell membrane. This is a slightly longer subtopic than last week, so it will take us roughly 3 weeks to complete. Similarly to last week, I've put the course specification on the first slide. Please have a read through this because it does describe all the essential elements of this subtopic and what you can be examined on. So we'll be looking at the cell membrane and its structure and what is meant by this term selectively permeable. Then we'll move on to passive transport. So there's two examples of passive transport that we focus upon, so diffusion and osmosis. Within that, we'll also look at what's meant by a concentration gradient and how that helps diffusion and osmosis to take place, and why it's really important for organisms to be able to do these processes. You'll also look at what cells look like when they've undergone these processes, either with molecules or fluid moving out of the cell or moving into the cell. Finally, we'll look at active transport, which is slightly different to passive transport and is used for different reasons. First of all, this week you should look at these 4 questions. These are questions that you should be able to answer from previous subtopics and previous science work in S1, S2 and S3. Jot down the answers in your jotter and then see if you can answer and mark these as you go through the PowerPoint. So like I said, this subtopic is going to take us 3 weeks. This week we're going to focus on the theory and looking at the different transport processes. So we'll look at osmosis, we'll look at diffusion and active transport. To start off with though, we're going to look at the structure of the cell membrane and what it's meant by selectively permeable. So let's first look at the cell membrane structure. All cells have a membrane which allow molecules to enter and exit the cell. It is selectively permeable, which means it allows only small insoluble molecules to cross. The cell membrane comprises of 2 main components, the phospholipids, which are these green items here, and the proteins, which are in purple. The phospholipids are arranged in a double layer with proteins interspersed throughout. So you can see that in this diagram. There's 2 layers of the phospholipids, and then the purple proteins can either sit within one layer or travel right through both of them. Because the cell membrane is described as being selectively permeable, it means that some small molecules, such as oxygen, pass directly through the phospholipid bilayer, while other larger molecules, such as glucose, are transported across the cell membrane by proteins. So we can see that in this diagram. Our carbon dioxide, similar to oxygen, passes directly through the phospholipid bilayer, but the glucose molecules have to travel through this protein, which lies through the 2 layers. There are 3 processes that we're going to focus on this week that allow substances to move in and out of the cell. The first 2, diffusion and osmosis, are passive processes. This means that it does not require any energy to take place, whereas active transport does. Diffusion is the movement of molecules down a concentration gradient from a higher to a lower concentration. It's an example of a passive process as it occurs down that concentration gradient and does not require energy. Diffusion allows cells to gain useful raw materials such as oxygen and glucose. These useful materials diffuse from a higher concentration outside the cell to a lower concentration inside the cell. So if you look at this diagram here, we have area 1 and area 2, split down the middle with this little line. So area 1 has a high concentration of these molecules, whereas area 2 has a low concentration because there's less of the red molecules. This means that the molecules in area 1 will travel into area 2 where there's a lower concentration. This will happen until there's an equal concentration on either side of the cell. The molecules actually continue to move, but do so equally in both directions, so there's still a net equal concentration. A concentration gradient is the difference in concentration of a substance between the two areas. A concentration gradient exists between a cell and its external environment as a result of an unequal distribution of molecules across the cell membrane. So like the diagram below, there would be a higher concentration of molecules on the outside of the cell than the inside of the cell, then molecules will move into that cell. If there's more molecules inside the cell than outside the cell, then the molecules are going to move out of the cell. Osmosis is a special type of diffusion involving water molecules. Osmosis is the movement of water molecules from a higher water concentration to a lower water concentration through a selectively permeable membrane. Osmosis is an example of a passive process as it occurs down a concentration gradient and does not require energy. So we can see in this diagram that we have a higher water concentration in solution 2 than in solution 1. So the water molecules are these red molecules with the two white hydrogens joined to the oxygen. As you can see in solution 1, there are more water molecules than in solution 2. This means there is a higher water concentration in solution 1, and therefore the water molecules will move into this lower water concentration until both sides become of an equal concentration. Osmosis affects animal and plant cells differently. An animal cell placed in a solution with a higher water concentration compared to its cytoplasm, such as pure water, will gain water by osmosis. This will cause the red blood cells to swell up and burst. An animal cell placed in a solution with a lower water concentration compared to its cytoplasm, such as a strong salt solution, will lose water by osmosis and shrink. So we can see that in this diagram where the cells have been placed in a higher water concentration compared to their cytoplasm. The water has entered the cell and caused the cell to fill up and burst. When placed in a lower water concentration, such as concentrated salt, the water will move out of the cell and that will cause the cell to shrink and shrivel. The effect of osmosis on plant cells is slightly different. A plant cell placed in a solution with a higher water concentration compared to its cytoplasm, such as pure water, will gain water by osmosis and become turgid. A turgid plant cell has a full vacuole and the cytoplasm and cell membrane push up against the cell wall. A plant cell placed in a solution with a lower water concentration compared to its cytoplasm, such as a strong salt solution, will lose water by osmosis and become plasmolysed. A plasmolysed plant cell has a small vacuole and cytoplasm, and the cell membrane starts to pull away from the cell wall. So the main difference for plant and animal cells is that you can see that when water moves into the cell with a plant cell, it does not burst like the animal cell did. There are also different terms which you use for animal and plant cells when describing the effect of osmosis. The main key terms here that you need to learn are turgid and plasmolysed. In task 1, you should copy the diagram on this slide, which shows the effect of osmosis on different plant cells. Using the information on the previous slide, Scholar or your summary notes, answer the questions here. So what way will the water move into or out of the cell? Describe what happens to the cell because of the movement of water, and write down the words used to describe the cells. For example, which ones are turgid, which ones are plasmolysed. Use the PowerPoint rather than a video to complete this task. The next task is very similar, but we'll look at animal cells and osmosis instead. So you're doing exactly the same, copying the diagram and then reading through your information to answer the following questions. What way will the water move into or out of the cell, and describe what happens to the cell because of the movement of this water. This allows you to have some comprehensive notes in your jotter to look back on and for revision. There are also 3 clips here that I would like you to watch. They're very short, so won't take much time, and they show you the effects of plasmolysis. So the first one looks at red onion cells, the second looks at red blood cells, and the third looks at why osmosis is so important. So our final process is active transport. So whereas diffusion and osmosis were passive transport, this is active transport. And active transport occurs when molecules and ions move from a lower concentration to a higher concentration. So this movement occurs against the concentration gradient, and therefore it requires energy. So it's kind of like, I always think of it as if you're walking up the stairs at school, that requires more energy than going down the slide. So active transport also requires membrane proteins to facilitate the movement of molecules and ions across the cell membrane. So remember before we looked at the glucose and the carbon dioxide, and the carbon dioxide was able to pass through the phospholipid bilayer because it was selectively permeable, whereas the glucose were using the proteins to get through. So one example of active transport is the uptake of ions by root hair cells in plants. And ions are found in a lower concentration in the soil water compared to the root hair cells. Plants must therefore use active transport to move those useful ions from a lower concentration in the soil water to a higher concentration in the root hair cells. So task 3 is this Venn diagram, and it looks at all 3 processes of transport that we've looked at this week. So we've got diffusion, osmosis, and active transport. What you need to do is add the statements here 1 to 12 into the correct segments of the Venn diagram. So for example, if one of the statements only refers to active transport, then you would put it in this area of the Venn diagram. Whereas if a statement applied to 2 of the processes, for example, diffusion and osmosis, you would add it to this section of the diagram. If one of the statements applies to all 3 types of transport, then you would put it in this middle section here. You might have to draw this quite big, so I would take a full page of your jotter at least, so that you have enough room to fill the Venn diagram in. Another good way to do it would just be to draw the Venn diagram, but also copy this table here. Therefore, you could just put the numbers in your Venn diagram, and it might look a bit neater than trying to fit all of this into the Venn diagram. So to summarise this week, we've looked at a few key points to start us off in 1.2, transport across the cell membrane. So first of all, we looked at the cell membrane and its structure and how it consisted of phospholipids and proteins. And we also looked at how the cell membrane was selectively permeable and what that meant. Then we looked at passive transport and how that occurs down a concentration gradient and does not require energy. Examples of passive transport were diffusion and osmosis. Diffusion was the movement of molecules down a concentration gradient from a higher to a lower concentration. Glucose and oxygen diffuse into the cells from a higher concentration outside the cell to a lower concentration inside the cell. Carbon dioxide diffuses out of cells from a higher concentration inside the cell to a lower concentration outside the cell. Osmosis was the movement of water molecules from a higher water concentration to a lower water concentration through a selectively permeable membrane. We then looked at animal and plant cells and what happens to them when they're placed in solutions with higher water concentrations or lower water concentrations than their cytoplasm. Here we looked at how animal cells swell up and burst or shrink, and how plant cells become turgid or become plasmolysed. Finally, we looked at active transport and how that requires energy for membrane proteins to move molecules and ions against the concentration gradient. So the tasks you have to complete are summarised here. First of all, you had to watch this video. Secondly, you need to read through that course specification. There are then 3 tasks that you need to do in your Jotter to ensure that you have sufficient notes. Finally, there will be an assignment on your class team, which will be the key terms and definitions for 1.2. This will be a multiple choice quiz. You will be given the definition and then have to select the correct term to match that definition. Finally, next week we're going to move on to focusing on the practicals that are included in this subtopic and that we would normally do in school. Luckily, one of the key practicals that we do in school can be done at home. I'm going to film this experiment, but if you want to give it a go yourself, I've listed the equipment here so that you can prepare yourself for next week.